Okay, this lecture's on models of cancer, and it's for the MSc Cancer Biology students and those studying the cellular molecular basis of cancer module. So in this session we're going to look at some of the models that we use to study cancer, both animal models and replacement in vitro models. I'm going to start off looking at models that focus on p53 and how p53 can be disrupted to induce cancer, particularly in experimental mice, and we'll look at whether these represent good models of human cancer. We'll then look at other tumour models where we use oncogenes and viral oncogenes to transform cells to turn them cancerous, and we can do this by looking at mouse models. So constitutive expression of oncogenes and viral oncogenes in mice will make cancer-prone mice. We'll also look at some Syngeneic models where these are spontaneous models where you've just got a cancer-prone colony of mice. And we'll also look at whether some of these constitutive models accurately represent the tumour in terms of the cancer stem cell hypothesis. And there's some argument to say that some of these models are not well designed to take into account cancer stem cells. So we'll cover that area. We'll then move on to using mice as xenografts for patient samples. So we'll look at simple xenografts where we use cell lines versus patient-derived xenografts where we take tumours directly from the patient and implant them into mice and then do our experiments on the mice growing the patient's samples. We call those PDX models, or patient-derived xenografts. And then we'll look at some more advanced in vitro methods of growing cells in a lab. So seeing how we can maintain tumour morphology using in vitro cell culture, and we'll look at see whether those are suitable models for doing experimental cancer therapeutics on patient materials. So we're going to have a brief look at organoids, some work I've done on protospheres, and patient-derived 3D cell cultures. And these are in vitro alternatives to patient-derived xenografts that are grown in mice, and potentially much more expandable and more widely available to patients if they work. So we start off with some animal models of p53. You should all know by now that p53 controls tumorigenesis, and if mice have two intact copies of p53, they live a relatively long life. If you engineer those mice to have one defective copy and one functional copy, they will live for a while and then will start to develop tumours, and most of this death is due to tumour formation. If you have animals which are what we call p53 null, they have no good alleles, they're both in this case deleted, and tumours start developing very early and you get a rapid death of the mice. Now this model here where we've got a good allele and a inactivated allele fairly closely represents human Li-Fraumeni syndrome, which is the inherited p53 inherited cancer syndrome. And this, you get tumour formation here because you have got inactivation of the second allele at some point during that mouse's life. And this follows very closely the Knudson's two-hit hypothesis of tumour suppressor gene inactivation. So you should all know that p53 is crucial for maintaining proliferation controlling proliferation and controlling apoptosis, and in response to DNA damage or some sort of stress or hypoxia, p53 will induce apoptosis and prevent proliferation, causing cell cycle arrest. What's less studied is the role of p53 in controlling quiescence, senescence and differentiation. So we know that in response to DNA damage, cells will become stressed, they will die, but this quiescence aspect is really important when we think about cancer as a disease of differentiation. So normal tissues have got stem cells, those stem cells differentiate into transit amplifying cells and then differentiated cells. And if p53 controls senescence and differentiation, then loss of p53 is going to be important with those processes. So we're going to look at examples where that occurs. So a really good model for looking at how p53 regulates stem cell behaviour is testicular germ cell tumours, because these tumours are generally p53 wild type. Now as this slide shows here, p53 is intrinsically linked in with stem cell markers. So stem cells are maintained by a whole bunch of stem cell pluripotency transcription factors, including NANOG, OCT4, SOX2, c-MYC, and KLF2. And you may well have come across these by studying induced pluripotent stem cells, because OCT4, SOX2, KLF, and c-MYC, if you transfect them into somatic cells, you will induce what we call induced pluripotent stem cells. Now, although NANOG is dispensable for the induction of induced pluripotent stem cells, we know that NANOG is crucial for keeping cells in a stem cell state. And p53 is intrinsically linked in with this because p53 suppresses NANOG. So if DNA damage is absent, NANOG levels can be high within the stem cell population, which is shown here. However, if DNA damage occurs, you get p53 stabilization by the pathways that you will have studied, ATM and MDM2 interactions and all of that type of thing. But activated and stabilized p53 will cause NANOG suppression. Now this is really important because once NANOG is suppressed, this embryonic stem cell or this stem cell can then differentiate. So in the presence of DNA damage, you can get p53 activation, and then a stem cell will cease to be a stem cell and will go on and differentiate, and then its progeny will stop growing. Whereas in the absence of DNA damage, you've got normal NANOG expression, and you can have a stem cell which will undergo self-renewal into two stem cells, or one stem cell and one cell that's going to go on and differentiate. So this shows that p53 controls cellular quiescence, and quiescence is this organised and deliberate lack of proliferation of stem cell-like cells. And this is shown here with respect to testicular germ cell tumours. These are particularly interesting because they are generally p53 intact, and they're incredibly sensitive to radiotherapy and chemotherapy. So it's not just down to the acute apoptosis induced by DNA damage. What we think is happening here is you induce some DNA damage with chemotherapy. That will activate p53. p53 will repress NANOG, and then we'll get differentiation of cells into proliferative cells, and then when they try to undergo the next cell cycle, that DNA damage will be recognised, and then they will undergo apoptosis because it will be recognised that they've got DNA damage. So if you follow this link here, there's a nice paper showing that chemotherapy depletes the OCT4 positive cells in an animal model of testicular germ cell tumours, and these testicular germ cell tumours are OCT4, NANOG and SOX2 positive, as you can see here. So if we're taking out all of the OCT4 positives, you're also taking out all the NANOG positives, and it's possible that those cells are all being taken out by activation of p53. Now, it is a general rule that once you get cells that are p53 deficient, they tend to become much more chemotherapy and radiotherapy resistant, because those cells don't respond properly to DNA damage signals, and also potentially normal differentiation pathways are altered. So on the previous slide you saw that p53 deficient null mice have a very short lifespan, and this slide actually shows extends out that type of experiment much longer. Seen here is even the p53 proficient mice will eventually die long term, and fortunately the axis is cut off there, but just 1 minute. Okay, we've now got the axis, so we can see that in mice, normal mice, once they get up to a couple of years old, you start to develop tumours, and mice start to die, whereas with the p53 heterozygous mice, Knudsen's 2-hit hypothesis at play, they develop tumours earlier, so you start seeing tumours after a year or so, and then the null mice, all the mice are dead well before a year. And what we can see with these models is that the range of tumours that you get an awful lot of lymphomas, both in old mice and p53 null mice, and in the p53 heterozygous mice you get a lot of sarcomas and osteosarcomas and soft tissue sarcomas. So this, to some extent, these mice do represent Li-Fraumeni syndrome quite nicely, because those patients also have a higher proportion of sarcomas. And this is summarised here. Our heterozygous mice have got one wild-type allele, one deleted allele, so they may represent Li-Fraumeni syndrome via a 2-hit model. When we look at the p53 null mice, we find that they have accelerated tumour formation. Now, this mouse model doesn't represent any human cancer model, as any human cancer condition, but it does really well allow us to study p53 roles in tumour formation. And what we find that these p53 null mice have a 3-fold higher level of these Lin1, SCAR1 and c-KIT positive cells, and that is the signature for hematopoietic stem cells, possibly accounting for the increased lymphoma that we see in these. So this shows that all of these mouse models have a potential use. The p53 wild-type mice, they'll develop tumours in old age, much like humans. Your heterozygous mice very much mimic Li-Fraumeni syndrome, and these mice are really used for studying what p53's role is in tumour formation, because many tumours that lose p53 become p53 null. So this has given us an insight into how p53 is altering the behaviour of those tumours. Now, what we've not covered are p53 mutants. So here, these are p53 heterozygous, where you've got one good copy, one deleted copy. Here we've got one good copy and one mutant copy, and these are generally missense mutations that change one amino acid into another, and these are what we call dominant negative mutations. And these are very interesting because many of the p53 mutations in human cancer are of this form. In some tumour types you do get tumours which are p53 null, particularly melanomas you will see that situation, and various other tumour types you'll get deletion of one copy and then deletion of a second copy. But the majority of p53 mutations or p53 inactivation is by mutant p53 with missense mutations. So p53 is really interesting because, unlike most of the tumour suppressor genes, p53 has a real over-representation of missense mutations, and this is looking at the frequency of different types of mutations in human cancer. Here's p53, and we've got 75% of inactivations are due to missense mutations, whereas you look at something like APC, you've got lots of frameshift mutations, same with ATM, same with BRCA1, lots of frameshift mutations, and in all cases a few nonsense mutations, so those are stop codons, and then there's always a few splice site mutations as well. So it's a really unique feature of p53 that there are so many of these missense mutations. And the thing about these mutations is they are dominant, and we call it dominant negative. They're dominant in that you only need one mutation. You don't need the second hit that you see when you've got a frame shift or a deletion. So where you've got a frame shift mutation, you mess up the reading frame and that's no longer functional, but you need the other good copy to be removed. With these missense mutations, they are dominant negative. So they are negative in that they are inactivating, but it basically means that the good copy is inactivated by the bad copy. And there may be some element of that going on over here as well, but we'll not cover that today. And this is maybe simplistically illustrated, but it shows what I mean. This is the dominant negative inactivation of p53 via a one-hit, almost oncogene-like mechanism. And this is all because p53 in its active configuration is a tetramer, and we've got 4 subunits. Now if you imagine a cell as producing 50% of its p53 is mutant and 50% is wild type, so wild type being blue, mutant being red, if you assume that you need 4 intact wild type subunits to have functional p53, then just by simple mathematics you would see that 50% mutant, 50% wild type p53 would actually result in 15 out of 16 tetramers being inactive. All of these are inactive, this one is active. So although we have got a reduction of total wild type functional p53 subunits by 50%, we've actually reduced p53 levels that are functional by 94%. That's 1 out of 16. However, to add to the complexity, these subunits, these mutant subunits, tend to be much more stable than the wild type ones, and that further adds to the excess of inactive subunits and a very, very minority of functional subunits. Now, that previous slide was probably a bit simplistic because that assumes that all the gain of function mutations, that's what these GOF, gain of function dominant negative mutations, it assumes that all those mutations are doing is taking out the wild type p53. And we know that that's not strictly true. Some of the gain of function mutations of p53 have direct biological action. So we know that if we transfect some p53 gain of function mutations into p53 cells, that should be p53 null cells, we increase colony formation. So it suggests that these mutants are actually having a biological role, much like a oncogene mutation. And sometimes we find that these p53 mutants have limited p53 response functions. So for example, a common mutation of p53 in humans is the arginine to proline 175, so that's amino acid 175. It should be an arginine, and it's now a proline. The equivalent mouse mutation, R172P, halts cell cycle but doesn't induce an apoptotic response. We know that p53 null mice will develop tumours at 5.5 months versus 11 months for this particular mouse model, so this is a less aggressive model, so we're not dealing with a completely p53 null phenotype by having this dominant negative mutation within there. What we also find is if you delete p21 from this mouse model, they get tumours at about near enough 5 or 6 months. Which is very similar to the p53 null mouse model. So that suggests that if you've got this mutation and you've got you're still getting p21 mediated responses, but if you then have a mouse that has got this mutation and the p21 deletion, you end up with a phenotype of no p53 mediated effects and the same sort of rate of death as the p53 null model. So this confirms that some of the dominant negative p53 variants have some p53 biological activity, and it's not just total loss of p53 function. Now, if we want to have mouse models that have p53 removed or inactivated to study, to induce tumour formation, we can do that by having transgenic mice that have been bred to have no p53. But we can also do other things. We can use SV40 large T antigen. Now this is from the simian virus. So SV40 large T antigen is a viral oncogene, and that will bind and inactivate p53. It will also inactivate unphosphorylated PRB, and that SV40 large T antigen can transform normal cells into transform cells, not necessarily fully malignant, but they will be transformed. They will have excessive proliferation, and they'll have attenuated cell cycle arrest and DNA repair processes. So this is shown here. The SV40 large T antigen is inactivating PRB and other related proteins, and is inactivating p53. And you should have covered this pathway extensively in previous lectures, but this single large T antigen is very transforming. Alternatively, we could overexpress the HPV E6 oncoprotein, which degrades p53, so same role here, or express the HPV E7 oncoprotein, which inactivates PRB there. So the combination of E6 and E7 is very similar to the effects of SV40 large T antigen. And then we can also express mutant forms of oncogenes. So something like RAS with a G12V, which has glycine codon 12 is changed to a valine. That puts it in a constitutively active form that permanently signals proliferation. So this is transforming, and what we often do is combine something like E6 overexpression with constitutive expression of RAS, and that will also transform cells into malignant-like cells. And this has been done in this particular study. This is a study I was involved in many years ago, and this is just the method section from that paper. And what we wanted to do here is get some cells expressing specific isoforms of vascular endothelial growth factor. And there are 3 different isoforms, and what we wanted to do was generate tumours from that only expressed 3 different individual isoforms of VEGF. So we did this by having access to some mice that were transgenic mice that had previously been designed by somebody else that only expressed VEGF 120, 164 and 188. And these 3 different mice plus the wild type mice were used. So by taking the mice at 13 days post-fertilization, took mouse embryos, got some fibroblasts from those cells, and then we took those fibroblasts and transduced them with the SV40 large T antigen, and we also put in a mutant RAS. So the large T antigen is depleting your P53 and PRB, and the mutant RAS is also driving proliferation. And in doing that, we used the cells that we isolated. They cultured them in the lab. They were now tumorigenic, so that they could be implanted into animals, and that was a sort of an animal model of fibrosarcoma, which is what these cells turned into. We were then able to use these tumours implanted into mice to show differential effects of VEGF 120 expressing fibrosarcomas versus VEGF 188 expressing fibrosarcomas. And this is just showing that it's using intravitreal microscopy. I'll not go into that. If you've done the translational research module, I covered that intravitreal microscopy there. But basically what we're doing is looking at the vascular formation of tumours. So the tumour planted behind the glass window underneath the mouse's skin, and these are the blood vessels, and then we treated them with combretostatin, which is an antivascular drug, and the tumours expressing VEGF 120 had this big area of where the vessels are being effectively created a large patch of hemorrhage, which didn't happen in the VEGF 188. So using this approach, it allows us to interrogate tumour blood vessels in a mouse xenograft system, but we're asking the question, what is the impact of VEGF from the tumour cells, not from the mouse? So we can do studies like that, but what we had to do is make the tumour cells ourselves from some mouse embryonic fibroblasts that only expressed these isoforms of VEGF. But these are the sorts of things that cancer biologists routinely do. Now we can take these oncogenes and dominant negative P53 mutations and drive their expression in all cells within the mouse. However, because they're dominant in activity, they're usually lethal. So you can't just create a KRAS G12V mouse because it will not develop. What you have to do is perform somatic activation of the oncogene, and that basically means that the mouse develops embryo normally, but you have a genetic construct in the mice that can be switched on only after the mouse has developed, because if you have this or this or this active during development, the mouse will die in utero. So you let the mouse develop properly, the mouse is born, and then you have a genetic construct in the mouse that is silent until you add something that switches on this construct here. And there are various things that you can use. You can use express SV40 or E6 or E7 under a tetracycline responsive promoter. So add tetracycline to the mouse and that switches on SV40. Or you can do conditional knock-ins, which I'll mention in the next slide or so, that basically can switch on the oncogene at will in somatic tissues. And in this particular model, you've got your wild type mice, you've got your percent survival, and then these are 2 various RAS mutant lines, and you can see you've got rapid tumour formation in the RAS overexpressing lines, and this just represents the different tumours that are formed, mostly lung and lymphoma and some general papillomas. If you follow this link, you'll get more detail on that model. So on that previous model, it was quite crude compared to what's routinely done now. In that previous model, you're adding a drug that will activate the gene expression construct, which switches on the oncogene. What we now tend to do is knock in transgenic models where you only express the oncogene of interest under a tissue-specific promoter. So for example, this study, knock-in of the SV40 oncogene into the prostate. So all you need to do this is express your SV40 in a transgenic mouse model under a prostate-specific promoter. So these mice will develop normally because the prostate is not, you're not completely altering the way that the mouse develops. You're just expressing SV40 in all prostate cells. Similarly down here, thymic hyperplasia and lung carcinomas in this transgenic line where E6 and E7 are being driven from a keratin 5 promoter. So these mice are developed with a keratin 5 promoter driving the reading frame for E6 and E7. That limits the cell types where E6 and E7 are expressed, and you end up with tumours in a limited tissue range. Now, for those who've done the translational research module, you may well have studied the science behind these conditional knock-ins. I'm not going to go through the technology, but if you do want to know how it works, search for CRE-LOX-P and FLOX-P sites, and you will find lots of resources on how these are done. So I'm not going to go into the technology specifically. So when we think about animal models of cancer, we want to know that those models represent human disease. And in previous lectures on this module, I've talked extensively about the cancer stem cell hypothesis and how cancer stem cells may, or possibly maybe not, mediate treatment failure. But all of these models need to take into account the cancer stem cell population if cancer stem cells are important, and if we're going to take them into account, we need them to be part of the model. Now, there are some models where this may not occur. So one model of prostate cancer is the what we call the TRAMP mouse model, and this is the induction of SV40 under the expression of a probasin promoter, and this is an androgen responsive promoter expressed in certain primitive prostatic cells. And we know that SV40 large T antigen will decrease P53 and PRB levels in those cells. So this androgen responsive promoter, which is prostate specific, expresses SV40, which is going to deplete PRB and P53, and that should create tumours in the prostate only. And indeed, on this model, that is what happens. Mice have PRB and P53 decrease only in these androgen responsive cells, and they develop adenocarcinoma very reliably. However, one issue with this is if your promoter driving the oncogene is not expressed in the stem cell population, then you are effectively bypassing the stem cell population. And this promoter here is more highly expressed in the differentiating cells than the stem cells. So effectively what's going on, or potentially going on, is that SV40 is only being expressed in cells as they cease to become stem cells. So you get all of your mutations and your abnormal proliferation in your differentiated or differentiating cells, but your stem cells may actually be completely untouched by this construct. Now similarly, there's this model called the EMU-MIC model. There is a paper by Kelly, you can find it here. This is the title of it. You can also find the entire paper here and here. This is one of the best papers for you to read because it is literally one page. That's the entire paper start to finish. And in this paper, it used the EMU, which is the immunoglobulin heavy chain enhancer, highly expressed in certain hematological cell lineages. So this EMU enhancer is driving the MYC oncogene, and in this transgenic mouse you get MYC expressed wherever the EMU IGH enhancer is expressed. So these mice develop B-cell malignancies, and there's also an EMU RAS that develops T-cell malignancies as well. Now in this paper, the attempt was made to prove that cancer stem cells don't necessarily drive cancer. And they did this because the traditional view of cancer stem cells, and especially if you follow the work of El-Haj, who were the first ones to really show the cancer stem cell population being a rare, maybe 1 in 1,000 cell. So it's a rare cell that has all of those special cancer stem cell behaviours. If that was true, you would have to inject at least 1,000 or 10,000 cells to get one tumour engraftment. And what Kelly showed using this model is it didn't matter if you injected 10 to the 5, 10 to the 3, 100 or 10 cells, you always got tumour formation, showing that cancer formation is not down to some rare cell like a classic cancer stem cell. It is any cell. It's almost like the stochastic model that I talked about in the cancer stem cells lecture, where any cell can transmit cancer from one mouse to the next mouse. So this is just the data here showing the tumour take frequency when you inject 10 to the 5, 10 to the 3, 10 to the 2, or just 10 cells. And you see 2 out of 2, 3 out of 3 are all developing tumours. So much like my concern with the tramp model, here the mouse model only becomes tumorigenic when the emu-MYC transgene is expressed. Now that emu-MYC transgene is predominantly expressed when the cells go down the lymphoid pathway, when IgH is required or active. So this is well after the hematopoietic stem cell stage, whereas if you look at the papers by El-Haj, who one of the first ones to develop to identify cancer stem cells, they're very much interested in the hematopoietic stem cell population. What this model is potentially doing is only inducing the MYC overexpression once cells cease to be a stem cell, which may not accurately reflect what goes on. So basically we might have got a model here that effectively bypasses the cancer stem cell population. So you induce tumour formation in differentiated cells, so therefore maybe by definition they are much more likely to be clonogenic and be able to transmit cancer than something that is dependent upon a mutant hematopoietic stem cell population, which is more likely to follow a cancer stem cell model. This model seems to follow a stochastic model. Now, one thing I should note about these models, as I've talked about some criticism of them, one thing that's really nice about these models is they're transmissible from one animal to the next. And that's because within strains of mice that are very, very highly inbred, one littermate is genetically very similar to the next littermate, and therefore you can transplant cells from one mouse to the next, which is why these experiments, such as these, can be done. Now, we have specific models of cancer that we call Syngeneic models, where this is routinely done. So Syngeneic mouse models are typically cancer-prone strains of mice within a single strain, and then if cancer forms in one of those mice, you can transplant that tumour into littermates or any mice from that same strain, and they will accept that tumour, so you don't have to have immunocompromised mice. So we call those Syngeneic mouse models. And a really nice one is the 5T2, 5T33 model. You could follow this link here. But because these are immunocompetent, they have more of the features of human cancers. Immunomodulatory drugs may well work, whereas in xenograft models where you've got immunocompromised animals, those drugs can't really be accurately tested. But just to show how nice these are, this is a multiple myeloma from a human patient showing osteolytic lesions in the bone. This is a mouse's leg with the 5T2 model showing osteolytic lesions in the leg there. So it does represent human disease quite nicely, and you can just take one of these 5T2 mice, take the cells from that and transmit it to other mice from the same colony, and they will then develop cancer fairly reliably in a few months' time. So much of what I've been talking about has been generating cancer in mice, transplanting them in mice to study how the cancers behave. And in most of those models we are inducing cancer and then treating the mice with drugs to see what works. Now there's an inherent weakness there in that we're dealing with mouse cancer. To try and make it more human, we can do something called patient-derived xenografts. So xenografts are where you've got human cells injected into a different species, in this case mice, and we have to use immunocompromised mice, either athymic or SCID mice, and the others are available. So with patient-derived xenografts, we take a piece of fresh human tumour material and place it into an immunocompromised mouse. And this is shown here. So we take the primary cancer, inject it into a mouse, and we'll call this the engraftment stage. And then what we can do is transplant, wait till that tumour grows, take that tumour out and then transplant it into more mice. And that's sort of an expansion phase where we can build up lots of patient-derived xenografts, also sometimes called avatars, and then serially transplant them from mouse to mouse to mouse to build up a large population. And now there's a huge database of patient-derived xenografts that you can buy. So these are mice. You can buy the tumour cells frozen down. You can buy mice that will accept those tumours, and this allows you to study tumours that are fairly closely related to the patient rather than relying on old-style cancer cell lines. And we'll come on to why cancer cell lines are not particularly good later on. Now with all of these models where you take the primary cancers, you can either do heterotopic transplantation, that would be subcutaneous, intravenous or intraperitoneal, and they just getting the cells into the mouse and hoping they take somewhere. Whereas you can also do orthotopic transplantation, where you would take brain cancer from the patient and inject it into the brain of the mouse, lung cancer from the patient, lung cancer into the lung of the mouse, and so on. So the reason we think that PDX models, are patient-derived xenografts, also called PDX, are particularly good is because we know that cell lines have adapted to cell culture. So you get lots of genetic drift. You can also get cross-contamination of cell lines, but it's this genetic drift. It's that, do the cells that you are culturing in the lab in a tissue culture flask actually represent the tumour or the patient from which it came? And the general consensus is probably not. So that's why we are really interested in this type of model, and it also means that you can do experiments from a patient, put that material into a mouse, test the mouse, and that predicts what the patient would respond to. So this is shown here how the avatar testing or PDX testing works. You've got your patient with cancer. You would take that tumour out, implant it into lots of mice, give them treatment A, B and C. If treatment B works in the mouse, you give the patient treatment B. Alternatively, you can take the cancer cells out, put them into mice, expand them in mice, just like here, then take out the tumours, maybe grow them in culture for a little bit, and then do high throughput screening in multi-well plates, and that will then inform on what treatments are used for the patient-derived xenografts. So this is the direct route. This is a sort of more research-informed route. The end result is that the patient gets the drug that either cured the mouse or was predicted to work based on high throughput screening, and that's reliant upon taking the tumour, putting it into the mouse, expanding it, putting it into a high throughput assay, testing maybe hundreds of different drugs, and then using that information to inform what drug the patient receives. So this can be done either on a patient by patient basis, or we can use PDX models to inform the best treatment for a particular cancer. So what tends to happen in the research phase is you get a collection of PDX mice, basically carrying patient-derived xenografts from lots of different patients. We then test drug regimes, we see which ones work. If we get a response, that is, there's the vehicle and that's the effect of the drug. If we find something that works, then we can potentially apply that directly to the patient. Or we can do what we've just seen in the previous slide, which is take an individual patient, create a load of avatars, test dozens of different drugs, and then work out which one works and then give the patient that particular drug. Finally, we could take that tumour, do bioinformatics, so we can do next generation sequencing on that, find out which genetic changes predict which drugs should work, then give the patient-derived xenograft the drugs that are predicted to work based on the next generation sequencing, find the drug that works and give that to the patient. So in part A here, in B here, you're sort of not informed by the molecular profiling, and you may be giving lots of different drugs, almost like a bit of a best guess, whereas here you are using next generation sequencing to say, okay, the sequencing suggests that this particular drug will work, let's just test it in the mouse. If it does, we'll then give it straight to the patient. So there are some limitations to PDX avatar models. Firstly, we're dealing with immunocompromised mice, so any drug that relies upon an intact immune system is not going to be best tested in these sorts of models. So something like PDL1, you might struggle to get sensible results from that. This could potentially be overcome by what we call next generation PDXs, where the patient-derived xenografts are engrafted with human immune systems. So we call these humanized mice, and then you can engraft the patient's cells into those mice. But that's getting a little bit more complicated. One of the main problems is that many tumours don't engraft into the PDX models, and we've found this. We've tried to do this many times and got a relatively low 20-30% hit rate on this. Some groups are proposing to overcome this by generating induced pluripotent stem cells, so that would be transiently expressing OCT4, SOX2, KLF2 and MYC into tumour cells, de-differentiating them, causing them to undergo expansion, and then you've got many more cells to put into your mouse model, but this might add some artifacts in there. So the next possible way to go, if PDX has become too laborious to do on a patient-by-patient basis, is whether we can do in vitro avatars. And in vitro avatars are basically growing the patient cells in a 3D cell culture system that maintains a differentiation status, and then maybe we can get a better hit rate on growing these cells and testing a wider range of drugs. Now, there's a real problem with growing primary cancer cells in the lab, and that is that if you take out someone's tumour, put it into standard cell culture, the vast majority of them will not grow. And those that do grow will not necessarily represent the genotype of the tumour from which those tumour cells came from. So what we have to do is grow the cells in serum-free defined medium, what we call cancer stem cell medium. And this is one study that we've done fairly recently, where we've taken some ocular melanoma cells, grown them in standard medium, and this is actually a cell line, but the principle still works with primary material. And in the cell line, the cells grow on plastic like this, fairly spread out. Put them into cancer stem cell medium for a couple of months, and they form these things called melanospheres. And then you take them out of cancer stem cell medium, and they go back to growing how they were before. There's some melanotic cells in the centre. These are amelanotic cells. So you can get phenotypic plasticity going on here. But this basically shows how different the cancer stem cell medium is compared to the standard serum containing medium. And you tend to get 3D growth much better in these defined serum-free conditions. Now, one thing we found by growing cells in these conditions is we maintain the chromosome aberrations that are in the primary tumour better than with serum. So in these ocular melanoma, the tumours lose chromosome 3, they gain chromosome 8q. is uveal melanoma, that's the same as ocular melanoma. So that's the genotype of the tumour, and what we would like to see is that genotype being present in the cells that we grow. Now, we know that that doesn't happen in serum, and during my PhD I cultured uveal melanomas extensively, and I generated or worked on 4 uveal melanoma cell lines, all of these cell lines have got 2 copies of chromosome 3. However, they all came from patients whose primary cancers had lost chromosome 3, which is what we call the lethal genotype. So what it's showing is, although we are growing cells from that patient, the cells that happen to grow in the lab don't represent the lethal genotype. So, and it's not that we'd contaminated anything, we'd proven that these cells came from the patient that they thought they'd come from. They were also melanomas, in that they had GNA11 mutations, which are classic for uveal melanoma. But what is effectively happening is that we are selectively outgrowing the pre-malignant melanocytoma, or less aggressive malignant clones, and the most malignant clones just do not grow in serum. So we were actually inadvertently selecting out for the least relevant cells from that ocular tumour, whereas if we grow cells in this medium, the data that's following up from this paper is showing that the cancer stem cell medium selectively maintains the chromosome 3 loss and the chromosome 8q gain in these melanospheres much better than trying to grow them in serum. So getting the culture conditions is right to getting the right cells to actually grow in cell culture. And this whole story makes me doubt the validity of an awful lot of cell lines. And this point is hammered out here by this paper by Ince et al. From a few years ago in Nature Communications. And what they are showing is, here's a load of ovarian tumour cell lines that have been generated under their defined cell culture conditions. And this is the histology of the primary cancers. This is the histology of xenografts. So these are cell lines grown standardly in the lab and then implanted into mice. Here you can see you've got epithelium and mesenchyme. Here you've just got a mass of epithelial cells. So this does not represent this. Whereas if you grow the cells from the patient in these defined conditions, you get, and then implant those cells into the mice, you get something that represents the original tumours. And they've got a mixture of epithelial, mesenchyme, you've got glandular epithelium here, whereas you've just got a mass of undifferentiated cells here. And what this particular group showed is, using their fancy medium, this OCMI medium, you get far more population doublings than with RPMI, which is standard medium, and other standard mediums. But more importantly, they showed that using these OCMI, is just the medium that they used, when you compare the genotype of the tumour to the genotype of the cells that are grown in culture, the cells that they could grow in culture had very similar genetic profile. So this is just the genomic loss and gain profile. So this would be an alteration to chromosome 3, either loss or gain, doesn't matter which, but that was also present in the primary tumour. When you look at cells from the same patient grown in RPMI medium and serum, you get a completely different genomic signature, showing that the serum is selectively growing out one population of cells, but the defined serum-free conditions is selectively growing out cells that represent the primary tumour. So this is why I doubt the validity of an awful lot of cell lines. So what I've been doing with my research group for the past few years is working with the authors of the paper by Ince on the previous slide, and the company called Cellular, who make a lot of that medium, to generate a bespoke lung cancer model to basically mimic this study but for lung cancer, and create a series of cell lines that we can then use for a platform for patient-derived xenograft testing. So we use their conditions, generated cell lines specifically with RAS, RAF, PI3 kinase and EGFR mutant, and aimed to test them on our new testing platform. It turns out it's not as simple as you think it is, and for even in their defined conditions we had great trouble growing out the malignant cells, because the non-malignant cells always seem to prefer to grow, which is a problem. One way around this is actually to try to grow tumours from metastases rather than primary cancers. In primary cancers you've got normal cells, you've got pre-malignant cells, you've got cells which are malignant but not quite as aggressive, and then you've got the cells that you actually want, which are the cells that are going to kill the patients. If you're going to take cells from anywhere, take them from a metastasis, because if you've got let's say a bone marrow metastasis from the lung, if you take that those cells, you're only going to get lethal metastatic cells. However, unfortunately, we can rarely get hold of that tissue. Anyway, to cut a long story short, we tried to culture cells from lung, and we found it impossible to grow the EGFR amplified and mutant lines that we actually wanted, whereas the RAS mutant lines seem to grow no problem. But anyway, using this, using the expertise from this group that generated this paper, we generated 4 new lung cancer cell lines that very closely resembled the patient's, and then we used those in an in vitro avatar type testing platform, which we call the cancer organ dot platform. And this is just shown here. This is in a 6-well plate. This is a polycarbonate membrane with tiny little pores and allowing liquid to feed up from here. This is what we call an air-liquid interface model, because the lung is an air-liquid interface tissue. And basically we pipette the cells at high density on this membrane, and we let them grow, and they form 3D clusters of cells on the top of this membrane. And this is what they look like down the microscope. Each of these are 3D cell clusters represented in probably about 100 cells. And then what we do is we treat these with drugs, and we see which drugs work, and then relate that back to the genotype of the patient to see if that matches. And this is just an example of a couple of patients that have been treated with a whole range of different drugs. Staurosporin is a positive control. This is a nuclear export inhibitor. We've got cisplatin, vemurafenib, selumetinib, ceritinib, and then some combination treatments. And what we can see from here is that staurosporin always kills everything, and in this case ceritinib works very nicely there and there, and KTP185 works in this one. That's a nuclear export inhibitor, but doesn't work in this. And this made sense because this was RAS mutant, this wasn't, and these drugs are being proposed to work in RAS mutants. So we've got something that seems to work quite nicely. So that organ.dot platform is just one of many different 3D cell culture platforms for growing primary cancer cells in vitro, and organoids are probably the gold standard. And if we look at the work by Hans Cleavers, a couple of links here, Hans Cleavers' group has really helped to pioneer all of this. He initially identified the stem cell population in colon and GI tract tissues, and then showed the cell of origin of many of those cancers. So have a look at some of those papers. Around the same time, we were working on something called prostospheres. These are prostate cancer spheres grown in vitro in a gel-like matrix, and there is a prostatic duct isolated from primary prostate cancer. And what we did here is selected the most stem cell-like cells from prostates on laminin, selectively cultured them on laminin initially, and then showed that these cells had very, very high levels of cytokeratin, which was classic, CK19, which was classic for what we expected to see, and we all ended up with things that represented an organoid-like structure that had variable prostate stem cell antigen, sort of focal points of that, various different markers. CD44 is always associated with the cancer stem cell population. So basically showed that these little clusters of cells very closely represented prostatic ducts, and that these potentially could be used for in vitro drug testing. Okay, so finally, it's really worth checking this link here. This is a really good review article on a group that has done extensively patient-derived 3D organoids for in vitro personalised drug testing. But this review really summarises the current field and the things that can be done. So classic organoids are taking a single cell suspension of primary cells, put them into something like Matrigel, which is a reconstituted basement membrane, put them in defined medium, and you get little spheres that represent, let's say, prostatic ducts or cancerous prostatic ducts, as shown here. So that's one simple way of doing it. You can make that a little bit more relevant by doing organotypic culture, which is where you take the epithelial cells and the connective tissue cells from the same tissue, mix them together, and you get reorganization, and you get you get the mesenchyme controlling the epithelial cells, so that works nicely as well. We can also go for tissue slices, and a lot of work's been done recently taking core biopsies, slicing it into small pieces, embedding it in some sort of matrix like matrigel, or sticking it on the glass slide, and then directly treating the drug, putting the drug onto the tissue slices, and then visualizing whether those live or die. And if you can get 30 tissue slices, you could test 30 different drugs. They're okay for short time point experiments. And then finally, probably the most relevant model is taking that tissue biopsy, cutting it up into small pieces with intact mesenchyme and epithelium, and doing something like what we've done here, but do it on a piece of tissue level, embed that piece of tissue in something like matrigel, have this defined cancer stem cell medium in there, and then we can grow little cancerous ducts of epithelial tissue. And this can be done also for mesenchymal tumours, but basically you've got the cancer cells with the normal support cells, and then potentially use this for drug testing. So follow up this review, because this really summarises the current state of this type of methodology. Okay, so that's it. We've covered everything from designing animal models of simple p53 mutations, right through to knock-in models, and then moving on to patient samples being grown either in mice, and then finally moving on to patient samples being grown in cell culture. So I think we've covered a fairly broad spread of what current cancer models are out there, and very much focused on using these models in towards the end of the lecture, using these models in personalised cancer medicine. Okay.